I'm curious. I am curious. Curious.
By nature.
Welcome to Curious by Nature. I'm Jessica Johnson from Newswise. There's a lot of self-help content these days and research that backs it up, encouraging people to reduce the negative effects of stress on our life. Did you know that high levels of persistent stress and traumatic experiences can have lasting effects on our DNA? Researchers at the Human Genetics Lab are asking, how does stress affect us genetically? Studies have shown that traumatic events like famine and war have the potential to literally alter people's DNA. Today, we discuss what they have learned about the subtle changes to our genes in the aftermath of violence and natural disasters. Dr. Mulligan, welcome. Please introduce yourself.
I'm Connie Mulligan. I'm a professor of anthropology and the Genetics Institute at the University of Florida. So I'm curious about stress, how we process stress, how we perceive stress, how we respond differently to stress.
Can you tell me more about your work overall?
I became really interested in stress. We all experience stress, but we all respond differently to stress. So two people can experience the same event, and one may come away from it, not even really registering the event, and the other one may be incredibly stressed out by it. So I'm just really interested in that variation in responses and what it says about how we've evolved to respond to stress. And then also what we can learn from those different responses to stress. So maybe we can learn to respond more productively to stress. So the main focus of my research is looking at possible molecular mechanisms that mediate the impact of stress on our health, both physical and psychological. So we know that too much stress is bad for our health, but how exactly does that work? And more, interestingly, how can it affect us years and decades into the future and even future generations? So as a geneticist, I started thinking about possible genetic mechanisms where there might be some way that the genome could embed these memories of stress, and that would help explain how these stressful events can have ramifications on us and our health far into the future. So, and that's how I became interested in epigenetic variation. So epigenetic changes are changes to the genome, but they don't actually change the DNA sequence, although they do affect how genes are expressed, how much of gene product is made, and they influence different characteristics about ourselves.
What is the difference between like a gene change and an epigenetic gene change?
So a gene change, so our DNA sequence or our genome is A's, G's, C's, and T's, 4 different bases that are just arranged in a string, and that's our genome. Those are our chromosomes. That's where what genes are. Actually, in the human genome, genes comprise a very small part of the genome. The rest of the genome, we're actually sort of still discovering what all it does. So we think of evolution as changes to the genome, but that's really pretty slow adaptation to the environment. Whereas epigenome changes, and particularly one type of change that I look at, which is called DNA methylation, and it's literally putting a methyl group, which is 1 carbon and 3 hydrogens, on a particular base of the DNA sequence. So the DNA sequence itself hasn't changed. It's just we got these methyl groups stuck on particular parts. And that methylation then affects how genes are expressed. So it can have an effect on how the genome is expressed, and also different characteristics about us, our physical characteristics, our mental health, our physical health, all sorts of different things. Epigenetic changes are also part of how every cell in our body has the same genome, the same DNA sequence, yet makes a different type of cell, a different type of tissue. It's because different genes are expressed differently to make a different cell. And epigenetic variation and DNA methylation is part of all that, changing how genes are expressed to change different outcomes.
What does that look like in maybe like a real life or a practical way for someone?
I mean, I think most people aren't really aware of epigenetic changes. Like you might have had your doctor order a test for a genetic variant to see if you have a genetic variant that increases your risk for some kind of cancer or something like that. So I think people might be sort of aware of what genetic variance and genetic variation is, but they probably aren't really aware of epigenetic variation. So how does it affect us on a daily basis? I mean, the biggest effect is what I just said, that how the same DNA sequence, the same genome can make all these different cells and all the different tissues in our body. But what's really interesting about DNA methylation is that it changes throughout our life. So not only does it change between every cell and every tissue, but it changes throughout our life, and it actually sort of tracks our biological age. But the fact that it changes throughout our life, and there's evidence that some of those changes are responsive to the environment.
How a gene expresses itself with an epigenetic gene and that expression, could that expression be different in different generations?
Absolutely. So it's part of all this idea of human variation. So we've got genetic variation that makes us different. We've got epigenetic variation that makes us different. So how different people respond differently to stress could have to do with different epigenetic variants, but then it could also be like this feedback loop where you respond differently to stress because of some of your epigenetic variants. But then by responding differently to stress, responding more strongly or less strongly, that could then further change epigenetic variants. So I study stress, but I also study trauma as sort of a very extreme form of stress in my research.
Can you tell me about your recent research study on violence and its effect on genes for generations that was published in Nature's Scientific Reports?
Yes, we just had that paper published, and it's very exciting. This is an international collaboration. It was actually started by Dr. Rana Dajani, who is a professor in Jordan, and she is the daughter of refugees who fled to Jordan. And she started wondering if the trauma of having been a refugee could possibly embed itself in the genome, possibly for generations. And she knew about the history of war and massacre in Syria, and the fact that there had been many Syrian refugees who fled to Jordan. And so she designed this experiment that that we published in scientific reports. The idea was that we wanted to test for an intergenerational epigenetic signature of exposure to violence. So Rana also works with Dr. Catherine Panta-Brick, who's a medical anthropologist at Yale University. And Catherine studies risk and resilience and conflict. And she's worked in lots of different areas, at lots of different parts of the world that have experienced war and worked with refugees. And Catherine worked with Rana to develop different interventions to help alleviate stress and build resilience in Syrian refugees. So Catherine and Rana then asked me to join the team to bring my epigenetic expertise. So because of Rana's understanding of the Syrian history of war, we have this really unique research design. So we have 3 different groups of 3-generation Syrian families that all have different exposures to war. So the first group, it was a grandmother who was pregnant during the 1980 Hama massacre. And then we're looking at her and her daughter, and then 2 of the grandchildren. Then the second group, the mother was pregnant during the 2011 uprising and war. And we're looking at her and her 2 children. So to go back to the first group, the 1980 group, we then have the grandmother who had a direct exposure to war. Then the baby she was pregnant with had a prenatal exposure to war. And then the little oocytes inside the developing fetus that represents a germline exposure to war. So we've got all 3 generations there. So we had those 2 groups where we had the grandmother pregnant during a war and then the mother pregnant during a war. And then we had a third group of control individuals. And these were Syrians who had moved to Jordan, not fled to Jordan as refugees, but moved to Jordan before 1980. So they didn't have any war exposure. So it was a control group. And so that's why our study is really the first to test for an epigenetic intergenerational signature of war violence, where we also have a control group of unexposed individuals. In our study, we had the grandmother who experienced the 1980 massacre, and she directly experienced the violence. Then the baby she was pregnant with had a prenatal exposure to the violence because it was the prenatal stage. But then that developing baby also had little oocytes, what we call gametes. They either turn into eggs or sperm as a grown adult. And in our case, we only took daughters because we wanted that second generation to also have babies. So in this case, it'd be the grandmother's daughter who then had the oocytes inside her, and those are germ cells. So that would be the germline exposure, because that was then the third generation.
Are there certain aspects of genes that we know affect, I mean, like colors of the eyes or things like that? I mean, that you could say, okay, it's on this gene, it probably, maybe there's an effect where I'll know where to look. Or is it fairly like a puzzle that it's kind of unknown and it's hard to tell?
So we know that there are about 20,000 genes in the human genome, and we have a function attributed to a lot of them. Most of the genes we know something about are genes that have something to do with disease, because that's how they sort of come to our attention, that something goes wrong and causes disease. With epigenetics, it's a lot harder because, like I said, only about 1 to 2% of the genome includes genes. But DNA methylation marks, like I said, there are about 15 to 20 million of them, and they're distributed throughout the genome. So there's this whole part of the genome that doesn't contain genes, but is certainly important. It's just we haven't figured out what it does yet. So when we look at epigenetic variation, we really don't want to focus just on the genes because we could easily be missing something. So we really looked at DNA methylation marks across the genome. We assayed 850,000 DNA methylation marks that is distributed all across the genome. And we tested each of them for an association with our different exposures to violence. So we had direct exposure, prenatal exposure, and then the germline exposure. And then we had to correct for all of these tests. When you assay this many markers, close to 1 million markers, you then have to statistically correct for having done all those tests to make sure you're actually identifying something that is statistically significant. So after all that testing and correcting and everything, we found 21 marks that were significantly associated with the direct exposure to violence, 14 marks that were significantly associated with the germline exposure to violence. So this is the first study to see those 2 intergenerational epigenetic marks associated with violence. And what's interesting then, if we look at that, we got 21 plus 14 marks, 35 marks altogether. 32 of them show the same sort of change in methylation, whether it's an increase in methylation or a decrease in methylation across all 3 exposures. So direct, prenatal, and germline. Even though they aren't all statistically significant, the 32 out of the 35 show that same change in DNA methylation, which really makes it seem like it's a common epigenetic response to war violence, or more broadly, stress.
Would you consider some of those changes as damages or just change?
It's really interesting to think about these changes and ask the question, are they adaptive or are they maladaptive? And we're not to that point yet. But the way I kind of think about it is that of all the DNA methylation marks we have on the genome, there might be just a very small subset of them that are responsive to the environment, responsive to psychosocial stress and war violence. And then there might be an even smaller subset of those that are heritable to future generations. And to me, it makes sense that those numbers would be quite small, because the majority of our genome and our epigenome has to continue functioning just like normal, even if I'm having a bad day, even if I haven't exercised, even if something horrible has happened to me. So from an evolutionary and an adaptive perspective, it makes sense that it would just be a very small number of DNA methylation sites that might be both environmentally responsive and heritable.
What is the difference between the genome and the epigenetic genome?
So the genome is the DNA sequence, it's our chromosomes, it's what people think of when you say genes. The epigenome, all of the epigenetic changes in one person then make up their epigenome. So genetics are just a small part of genes, and then the genome is the whole genome, all the DNA, all the chromosomes, in the same way that we can talk about individual epigenetic variants, and then all of them make up the epigenome.
So when someone is looking for like a gene variant, is that automatically an epigenetic?
No, most people, when they talk about genetic variants, they're looking at variable positions in the DNA sequence that is our genome, that is our chromosome. Epigenetic variants, particularly DNA methylation, are those little methyl groups that are stuck to bases in the DNA sequence. And they can come off and come back on and come off and come back on, but the DNA sequence stays the same.
Epigenome can be changing for a person constantly. That's why it's epigenetics, because you can, like, through even our actions, through our environment, we can change it, right?
Yes, yes. Well, and so that's a great question, because if we learn more about the parts of the epigenome or the DNA methylation marks that are responsive to the environment, can we then intervene and change them? So there's research out there studying things like meditation, um, spending time in green space, changes to a diet, to see if they can change those DNA methylation marks.
Right.
And so another interesting thing about DNA methylation, like I said earlier, it tracks our age. So we can calculate something called an epigenetic age based on certain of these DNA methylation marks. So we studied 850 DNA methylation marks, but there are about 300 to 400 of them that have been identified as being highly correlated with chronological age, and therefore you can use them to calculate an epigenetic age.
It sounded for a moment like even the person that was experiencing firsthand the violence, you would think that that person would have accelerated aging, right?
It's very hard to know what to expect. So yes, you might think that the direct exposure would be the most extreme, the most intense. But what's also true is that the fetus is at this very early stage of development, where they're very phenotypically plastic, meaning there are lots of different directions they can go in terms of their final phenotype or their final form, if you will. Whereas a born individual, and particularly an adult, is kind of fixed to a certain extent. There's just not that many different ways for them to go. So we thought that it might be this prenatal stage that's very responsive and very phenotypically plastic, that might be more sensitive to the war violence exposure as measured by epigenetic age acceleration.
Right. So that is very that's possibly another takeaway, is how important it is for pregnant women to be in healthy environments, right, for their child.
Absolutely. Absolutely. As if we didn't already know that it was really important that pregnant women, take care of themselves and their baby. The idea that stress and trauma that the pregnant woman experiences might actually embed itself in the developing baby's genome and in the germline cells, the little oocytes, the little sperm in that developing baby as well, has really profound implications.
This is really so interesting. Thank you so much for sharing more about it and going into more detail and explaining what everything means.
Curious by Nature is a production of Newswise. More than 7,000 journalists get the Newswise wires. Visit newswise.com and find out how to spread your news today.